And I just want to very quickly kind of overview and kind of provide guidance if anyone's a little bit lost in terms of how to interpret the data, and kind of go through the way I kind of logically think about projects and how we kind of bring all the tools that we've kind of learned about together in terms of the workflow that you can undertake to explore and interpret your data. So first thing I want to mention, it's, untargeted, non-targeted work. It still is important that you have experimental design. So, replication is good, making sure that you isolate your variables. But that said, there are many different use cases of using metabolomics, and all of them have different goals, objectives, and different tools that you would use to kind of fulfill those goals. And just very broadly, and by no means is this comprehensive, kind of cover two main uses that I kind of, if I had to lump everything together, and that's the question of characterization. So there you have very few samples. You may even have one samples, but you want to really do a detailed, in-depth chemical analysis to understand everything that's there, all the analogs. We've talked about a little bit of that today. So, some kind of example questions that you may ask. So what is the chemical composition of blood, right? There you could imagine you could just take one representative sample, like someone missed the standard reference material from NIST, and you could just go through, extract that multiple ways, use different types of chromatography, different types of mass spectrometry, try to all bundle it together to understand what's truly in the blood sample. Or more specifically, you can ask, what are the contaminants specifically in blood? And then you may, subset and do more of a exposomics type of experiment, which in my mind is kind of a subset on the greater untargeted or non-targeted metabolomics question. And kind of like I alluded to, a similar question, are there any kind of unique metabolite transformations in the blood, or in the case of like a bacterial culture, you may say, what is really unique about this bacteria, right? Without having any kind of outgroup or comparison. And then to kind of strongly contrast that, then you have, those are actually two different colors, Scott. Dark red, light red. He's colorblind, so I had to get a small joke in there. So, you have the comparison, right? A versus B. So the blood versus the skin, subject 1 versus subject 2. So you could generalize that, right, to pretty much anything. And of course, those are questions like, how is the blood in healthy controls different than persons with some X disease, right? Or you could take it a step further, kind of parallel in terms of the characterization of questions, what are the contaminants different, right? Are one set of group exposed to a different type of PFAS or to more PFAS, right? And then are there any kind of unique metabolic transformations? Individuals have different metabolic capabilities, and maybe one person is deficient in one enzyme that helps transform a drug or exogenous compound. That said, the kind of tools, by no means, again, a summary of what we've kind of covered, but in the characterization, I'd say the strength of a lot of the tools that we learned about kind of fall in this category, right? Kind of at the fundamental characterization of molecular networking, Sirius, deep score, MS2 query, MS2 LDA, all those things, understanding more about the data that you have, right? That's not to say that's not useful at all in the difference between groups. In fact, it's very useful, because after, and again, my argument would be, you're probably most interested in doing the statistical analysis between the two groups first to figure out what you kind of need to focus on. But then you always end up going back to the characterization part, right? Because you always have unknowns in the data. There's always these unannotated things that you need to know about. So learning the fundamental bits about characterization, I think, is super helpful. That's also why the majority of the course was kind of on that. So just kind of put everything in perspective. And again, you all will have the slides. This is kind of how I approach it and think it and use these tools. Again, high level overview. kind of a very simplified way of I kind of think about the interpretation, some guidance for you. So we talked a lot about the raw data being converted into open source. It's still very important that you evaluate and inspect your raw data. So open every file, make sure injected, make sure the pressure looks good. That you could do a little bit of this in some of the processing tools, which we saw, right? That MZMine, you look at the pressure trace, you can look at things like that. This is, again, very generalized. But we learned about MZMine for the feature finding part. We talked a little bit about like data transformation, so normalizing, scaling, transform. It's very important that you do this, but it's highly situational. So I would argue this is probably one of the hardest things to know, because you need to know a little bit about your data set. You really need to evaluate, and I definitely suggest that you look every step that you do to the data, you carefully evaluate the outcome of it, so you're not introducing any kind of shifts or bias into your analysis. I always like to start with the multivariate statistics. So just doing a simple PCA, right? Kind of like what we're doing today, or a PCOA, something that's unsupervised, that's you're really looking at the data structure, right? Not rely on any kind of additional information that you have, just to make sure that everything makes sense, right? Like in our case, blood kind of looks like blood, skin looks like skin. And then at that point, I then feel kind of confident to go down to the univariate statistics. We kind of talked about that. And then in the end, I think if I personally had a like takeaway message, it's just super important that you validate those results in the end. Be very careful about interpreting the p-value, like what test you're doing, what it means. A smaller p-value doesn't necessarily mean it's any more valid. 180 to the -6 is good. 180 to the -7 is also really good. It's not really better. It just means that there's a less likelihood that, it's a false discovery. This is all data that you all probably should have seen or generated by now. Just kind of overview. Again, with the multivariate statistics at the highest level, just applying some rational thought, right? The things that kind of should be highly differential should appear very differential. And just as an example, not all PCA plots will look like that, right? Blood and skin are very different. So we use kind of the apples to oranges analogy here. So they are very extreme. They should be extreme. Like, I think you would argue that you don't need a $500,000 instrument to determine that blood is not skin, right? But it's a good sanity check, right? Particularly including blanks or quality controls, right? So it's good. And then if you want more detail, of course, like PCA or PCOA, it's dependent on the data that you provide it. So if you want to see the differences between apples, right, it doesn't make sense to include oranges in that comparison. So you should subset the data and reprocess. And then just to explore all the factors that that are going on in the experiment. So that's kind of on the right-hand side, right? Again, there's two red colors there, Scott. Understanding like what the structure is, right? That there are, individuals, right, in this data set that do seem to cluster together, right? So understanding that, it kind of gives you indications that may be something that you need to account for in your end analysis, that there is kind of this individualized response. And as just to remember or reiterate, the p-value, right, and the Adonis R-squared value. So this is like, you could say subject is accounting for like 39% of the overall variance, right? So it's pretty significant. And then just to review, so like that's kind of the multivariate statistics. Univariate statistics, you could do it individual, kind of like Philip showed. Heat map is a good way to generalize that. So I think many of you have been able to generate the heat map, and we can definitely help out after this if you're struggling with the coloring or something like that. But then, then it's relying on you to look for the patterns, right? And people are pretty good at this. So you can kind of look, there's definitely chunks of blue and chunks of red. And, depending on, if you can read the legend or not, which you can up here, but you may be able to do on your own screen, it's ordered in terms of time, right? So you're kind of looking for these patterns that occur over time. So, maybe this streak of red is very interesting in the fact that it kind of is very low in the beginning, goes up, and then generally falls. So just again, just a high level overview. I think this is particularly nice because it's also predicting the compound class. So this is the top dendrogram. It's split out. You can kind of see at a high level that there is some similarity between compounds within the same class. So if you're doing a metabolomic study, if you see if you see differences between groups, but they also occur within the same kind of family or pathway, it kind of adds more biological weight, right, to your conclusion. Like it seems more likely to be true instead of having this spurious kind of results where it's like one compound per class, where there's seemingly no obvious relation biologically. By the way, this is very hard. So interpretation takes a lot of time. So I just want to kind of carry through and relate the heat map back to GMPS. So asking that question, and you can definitely emulate this in your own workshopping session here. How you relate this feature found in, say, the heat map back to then your characterization work, like your molecular networking job, right? So the nice thing about how it's kind of structured with MZ-Mine and all the tools that we've learned about as this conserved feature, right? That then you can relate all the way back to all these different tables. So you can go from your process plot all the way back to the original MZ-Mine table and even understand the particularities of the feature finding that what occurred, right? So this is typically done on the last part, right? Validating your findings, that you could kind of trace it all the way back. So like if we just use this particular feature, for example, you may not know what it is. I happen to know what it is, but let's say it's an unknown, so it was not annotated initially. You can then, before that, just to remark, so this is great for looking at the univariate data as a whole, but of course, then you can look at the time course analysis, and this is specific to this project, right? But you could do the equivalent like box plots, violin plots, or something like that shows you the difference between groups. And again, just to note that it's the same feature. And like if I was interpreting this, I would say something like, this particular feature is observed in plasma, but there's no evidence for it in skin. It's always good to say that you didn't observe it or you didn't detect it, rather than it's not truly being there. It just may be below the limit of detection, or it may be convoluted with another feature that's concurrently detected in skin or suppressed. So there's definitely some analytical things that kind of go into the measurement of this. And then you can remark on the time pattern, right? Like it kind of goes up over time, it peaks at a certain time and falls. Again, this is just kind of an example. And then you can write all that in a paper, but I would almost guarantee that the reviewer will ask, what is it? How do you know it's real? And then, it doesn't make sense, right? So this is a forewarning. If you remark on something that's unannotated, you may get a lot of pushback from reviewers. That said, sometimes that's just the limit of of what we do in terms of doing discovery work or hypothesis generating research. So if you need to find that feature, one way that you can find all features is in your GMPS link. It's that particular option. It's view all spectra with IDs. It says IDs, but just ignore that. As opposed to the first link in that option, which is view all library hits. That one gives you all the features that have like a library match. So it doesn't actually give you the full list of things that were unknowns. So if you click on that, it gives you the table. And just to orient people, again, these slides will be given to you, but that cluster index is the column header. That actually is the same as your feature number. And some of this is like legacy, it's called different things. So if you type in the feature number, you can find the exact feature that you're looking for in the MZ-Mine table. And just to remind you that that aligns with the MZ-Mine table that you get out in terms of the row. Or you can simply search by the precursor mass or retention time, since you have that information as well. So like in this case, I kind of subsetted it for you. I was looking, in this case, precursor mass, and I just made a very general range, like 432 to 432.5. I can find my feature that I've highlighted here. It has the right retention time, and it has the right feature index. So all those align. There may be a little bit of discrepancy in terms of the exact feature name and what's in the table, just so people are aware due to rounding. So like in the feature, it's 3.182 minutes. In the GMPS, it rounds to two digits. So, but the feature index will always be the same, or should be 3770. In this particular case, there's no button, and I forget who pointed this out the other day in the network browser. But if it has a network node, or if it's within a network, there'll be a link that says View Network right there. If you click on that, then it opens up the in-browser visualization. So this is kind of showing, reminding you of the things that you've learned previously, that you can go in and understand that there's connect nodes. If there's not a link there, then it does imply that this is like a singleton within the network, that there's not anything related to it. And there's any number of reasons for that. But in this case, it just means that it seems to be unique and off by itself. Pointing out two links that I think that we kind of glanced over. There's a lot of things in GMPS, but relating to tools that we've already used or talked about a little bit. So if you click on the button that says masked spectrum for that line, it will bring you to masked and already should auto-populate the job for that spectrum. So it's a shortcut. So then you can immediately take it and then search it against the database to see where it's seen before. So again, kind of a way to figure out where your compound is, what the annotation might be. So if it gets called up with other drug studies and diphenhydramine, maybe you're like, okay, maybe it's diphenhydramine related, or if it comes up with food studies, okay, maybe it's a food-derived molecule. And I didn't show you the masked page, launch page. It's actually migrated to GMPS2, which you kind of heard about with Ming. But I just want to remind people and call out the documentation for all these tools are online. It's pretty good. It's pretty comprehensive. So if you ever have a question, because there's no way that it was comprehensive in terms of what people covered in the conference, in the workshop, the documentation has a lot of details on everything that we've talked about. So I'll leave that for another day. And then this other button that says USI links. I don't think we've mentioned that either. I find this particularly useful. If you click that, another like box will pop up and it'll have this long name string. And it links out to another resource. And USI stands for Universal Spectrum Identifier. And the idea behind this is that it's a unique code associated with that exact spectrum, right? So your file, that spectrum specifically. So you can like take this, put it in a paper, and then someone can exactly find that same exact spectrum in your file in your open data. So I really like this concept. I've used it quite a bit. I should use it more, to be completely honest. And it has this web page where you can interact with it and you can actually change the plotting as well. So I like this because, you know, sometimes the plotting software isn't great for pulling out the MS2. So you can change like which peaks are have a value. You can change the scaling, where the intensity threshold is listed in terms of what's labeled. And then you can export it as different. I know it's kind of hard to see, but the blue buttons at the top are different file formats. So you can export it as like a SVG, which is like really nice for putting in the paper. Yes. This is for anything that you run in like a GMPS job. Every line will be generated a link if it has an MSMS spectrum. Yeah, so you can, even the knowns, unknown ones, you can basically put in the SI so then people can directly see why you annotated it as such, right? So you have to run it through GMPS. And I believe it works for, oh, that's a good question. It certainly works for public data sets. Yeah, so I think the data has to be uploaded to Massive, which is the common route of using GMPS. And I'm not sure. That's a good question.
No. So any
if it's
flow that runs through GMPS, generate the USI for it,
yeah.
and put that USI into the anywhere, and someone can pull that up and see that spectrum. It doesn't have to be publicly available.
Yeah. It's just that spectrum. It doesn't link to anything else.
Molecular networking workflow, it's recommended that you actually put the link into the paper. That link doesn't have to be to a public data set.
Mhm.
You get that single workflow, someone can download the graph ML if they can't rerun the data or
Mhm.
so the USI generated no matter what, private and public.
Thanks. And I think the only last thing to kind of call out, so this is like a spectrum that you can get out. And then, so being who I am, there's certain things that I look for in terms of patterns. Because I'm just like used to looking at diphenhydramine stuff. So I kind of immediately saw things that are reminiscent of diphenhydramine. So the diphenylmethyl cation, the 167 peak, you've seen that probably before. And then 256 is actually the parent mass, or it's the mass of the parent compound. So the actual loss from the precursor ion amounts to the neutral loss of the glucuronide sugar. So it was something I already kind of was looking for. It's kind of new. But if you didn't know what it is, if you had no idea, that's where some of the in silico prediction tools are really strong. Sirius, MS2LDA, things like that. So like in this case, like if you look at the MS2LDA result for this particular mass, which is hard to see, but it's the diphenyl methyl cation, the 167 peak, you could start to see that all the structures are wrong in terms of the complete structure, except for this one. But they all have that common subunit, right? So it gives you piece by piece information of what the structure can be. And unfortunately, this is like a pretty tedious operation, right? To kind of search through the structures, verify. But I thought it'd be important to kind of visually walk through the ways that, you can go about this. So I'm just going to leave this up for people while they work, just to remind you all. But I think I think that's it, unless anyone has any questions. Yeah. So I think we can continue to work in our groups, and then we'll maybe make an announcement as to when to start evaluations. Is that right, Fallon? Oh yeah. Okay. Yeah. So if anyone couldn't hear, if your group is kind of ready to present for the ETCS credits, is that right? Yeah, I'm familiar with that. So I'm glad I didn't murder that. Just let us know so that we're not waiting for people to finish when you could already be presenting. So thank you.